Interphase is the longest phase of the cell cycle, where a cell grows, carries out normal functions, and prepares for division by replicating its DNA in organelles. Interphase consists in 3 stages, G1, S, and G2. During G1, the cell grows larger and makes proteins and organelles. It also carries out its everyday functions. This stage helps the cell make sure it has enough energy and resources to continue. The DNA is spread out inside the nucleus as a chromatin, not chromosomes yet. The nucleus is visible, which means the cell is making proteins. The centrosome is also present to help prepare for the cell division later. S phase, which stands for synthesis, in this stage the cell copies its DNA. This is important because when the cell divides, each new cell needs to complete an identical set of genetic information. Inside the nucleus, the DNA is still spread out as the chromatin, but it is being duplicated so each chromosome will have an identical copy. The centrosomes are also duplicating to help prepare for the cell division later. Then comes G2. During G2, the cell grows a little more and prepares for mitosis. It makes the proteins needed for division and checks the copy DNA for mistakes. If there are errors, the cell fixes them before moving on. The DNA is already copied and stays loose as a chromatin inside the nucleus. The nucleus and the nucleus are still visible, and the centrosomes are present to help prepare for cell division. The cell is getting ready to enter mitosis.
Prophase. In prophase, chromatin begins to form into chromosomes. And as you can see, DNA then coils into sister chromatids. In the center of each chromosome is a centromere, which attaches the sister chromatids together. In the cytoplasm, microtubules, known as spindle fibers, begin to fan out of the centrioles. They then begin to move on opposite sides of the cell, making the nuclear membrane disappear. Now that the nuclear membrane is gone, the opposite ends of the spindle fibers can attach to the centromeres.
To summarize the metaphase stage, the nuclear membrane was already broken down in the stage before. Then the sister chromatid pairs move to the center where there's an imaginary metaphase plate. There's one microtubule on each side of the cell, and each kinetochore on each sister chromatid is attached to a microtubule, and the chromosomes are just waiting there.
Anaphase. During anaphase, spindle fibers separate the sister chromatids at their centromere. Once they are separated, the single-stranded chromosomes form a V-shape and move to the opposite poles of the cell toward their centrioles.
Last stage of mitosis, the 2 sets of daughter chromosomes get to the spinal poles and deconsent. Then 2 nucleus form. This usually means mitosis is over. After mitosis is done, it is followed by cytokinesis. The contractile ring of actin and myosin filaments form a rapidly deepening furrow and pinch the cells into 2 new cells. The ring contracts. There is a fusion of intracellular vesicles to add more plasma membrane to compensate for the extra surface area. At the center of the intracellular bridge, there is a mid-body that connects with intracellular vesicles to go on to abscission to create the 2 nucleus that finish cytokinesis.
What happens when the cell cycle goes wrong? This is the DNA replication. Once it is badly damaged, it can lead to cancer, tumors, and many other problems. For example, apoptosis, which is the next thing we will be talking about.
This is the DNA replication.
Apoptosis is when the cell is unable to repair damage and unable to pass the checkpoint, so it undergoes programmed self-death. This is a checkpoint, red checkpoint, obviously, right? This is our cell. So basically, the cell is gonna try to go to the checkpoint, but look what happens. Boom, boom, boom. Kaboom!
I need some wet shit, I need some bacon, yeah, I'm a little man.
And you never knew how much I really liked you, because I never even told you all I meant to.
Genetic disorder. When the cell cycle goes wrong, it could have an incorrect amount of chromosomes, which then leads to genetic disorders.